Alright guys, so this is going to be a video discussing our DNA replication notes. Once again, this is just for our academic class. This is just for our regular class. Honors, you do need to know this material, but you will have a video coming out a little bit later that will describe this in a little bit more detail. But it's always good to make sure that you know the basics before you can move on to the more complicated stuff. So DNA replication is really pretty basic as long as you understand the structure of the DNA. Now I do have a DNA structure video that you can watch before you watch this one. Because if you don't understand how DNA is structured, you're not going to understand how it can replicate. So the importance of DNA replication is that you need to be able to have a full set of DNA in every single one of your cells. And you really want that DNA to be exactly the same for every single one of your cells. If it's different somewhere in there, then that's called a mutation, and it can lead to cancer and birth defects and all sorts of other things. So when your cell divides, when you go from just one cell to two cells, you need to go from one set of DNA to two sets of DNA, and they need to be exactly the same. Now, if we remember and we think back to our DNA structure video, we said that each side of DNA is complementary to each other, so they follow base pairing rules. So what that means is you can create a new set of DNA using the old set as template. So this is called semi-conservative replication. Semi-conservative replication just says that when you go through DNA replication, you end with two strands that are one half old and one half new. So in this example, our old strand of DNA is in red here. What we do is we split it in half and we add in new blue bases here. So if you have just one side of it, that side is going to be half red and half blue. It's half old and half new. So that's called semi-conservative replication. Now there's only 3 steps for this. The first step is that you need to rip the DNA in half. And this is why those hydrogen bonds that we talked about earlier are so important. Those hydrogen bonds are very weak bonds, so it allows you to literally just come through here and rip or unwind this double helix all the way up through here. The next thing is that you're going to add in new complementary bases here. So the A's are going to be paired with T's, C's are going to be paired with G's on both sides. And so you'll do that for not only one of the strands, but for the other one as well. And then at the end, you fuse this new nucleotide backbone together. And so you end up with 2 strands, each that are half old and half new. So here's another example. This is going to be closer to what you do for your project. I'm going to upload a project instruction video here in just a little bit as well. So in this example, our bold is going to be our original strand, and our italics are going to be our new strand. So here we start off with just one copy of DNA. A is always pairs with T, G is with C, T is with A, C is with G, so on and so forth down the line. So the first thing that we're going to do is we're going to come in here and we're going to rip this DNA in half. So literally it's going to go from here where it started, and we're just going to kind of tear it apart. Once it's separated, you're going to move on to step 2, which is adding in the new nucleotide bases. And it's important to note that these are not connected by those phosphodiester bonds yet. You're adding them in one at a time. So nowhere on here are these bases connected in this row. But we still use our base pairing rules. A pairs with T, C pairs with G, T pairs with A, G pairs with C, G pairs with C, C pairs with G, so on and so forth. And you're going to do that for both strands. And the very last thing that you do is you fuse the new DNA backbone together using a strong covalent bond. Remember, more specifically, these strong covalent bonds are called phosphodiester bonds. They're just the sugar phosphate covalent bond, though. And so we can see that we have the same strand that we did before. The only thing is that we've added in those covalent bonds. The other thing that I want you to notice is that our top strand and our bottom strand are exactly the same. A, C, T, T, G, 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 C, A, A, T, T, G, 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 C, C, A, A. All of them are the same. You've created two new identical DNA strands that are 1/2 old DNA and 1/2 new DNA. Now, if you're one of my honors students and watching this, remember we do go into a whole lot more detail here, but for right now, this is a really good ground for you guys to kind of start at. If you have any questions, please send me a message on Remind or in some other way, let me know. All right, bye guys.